Welcome. Our next lecture is going to focus on viruses and prions. So let's get started. Viruses are infectious acellular particles that must invade living cells to replicate. Keep in mind when I say acellular, they do not have cells. Viruses are, in essence, particles that have to infect living cells to take over the cellular machinery, in other words, ribosomes, etc., to begin the replication process for new viruses. Therefore, they're called obligate intracellular pathogens. Now, on this table, again, it's like many of the other tables. I don't want you to necessarily memorize everything, but I do want you to go over it and get very familiarized with it. As we've said before, viruses are not cells. As a matter of fact, they're not considered alive. They are generally smaller than prokaryotes, and most require electron microscopy to see. To give you a better perspective, it was the late 1890s that viruses, evidence started to build for these organisms. They were filterable. They were able to be filtered out. But unfortunately, it wasn't until the mid-1930s with the development of the electron microscope that anyone was able to see a real virus. The structure is usually a protein capsid coating with a nucleic acid. The nucleic acid could be either DNA or RNA. Its replication is totally dependent on infecting another cell, and in that cell, taking over the cellular machinery and getting the cellular energy, ATP, to do the replication process. We'll get into more of this in detail in a minute. Viruses do exhibit diverse structural and genomic features. We need to go over some important terms here. With the term bacteriophage, these are viruses that infect only bacteria cells. Animal viruses infect animal life forms. Some of them will actually jump species to infect other species of animals than their original host. Case in point, COVID-19, where we first thought it was infecting bats and came to eventually infect not only human beings, but there's evidence that it's infected other animals, dogs and cats, domestic animals such as that. Okay? A virion is a single infectious viral particle. A capsid is the protein shell of the virus that packages and protects the genome. Capsid's shape depends on the shape and number of the three-dimensional subunits called CAPSOMEREs. When I talk about the term pandemic, I talk about a worldwide outbreak, otherwise known as a global epidemic. Now, the shapes of most animal viruses consist of one of these three types, or are basically categorized in one of these three types. Helical, so the capsid appears as a hollow tube. ICOSAHEDRAL, which is constructed from a hexagon and pentagon-shaped capsomeres. As a result, it may appear spherical. To help you to understand that, think about the shapes you see on the outside of a soccer ball. Now, complex, this is a catch-all term. It includes bacteriophages, the ovoid brick-shaped structure of a pox viridae, such as smallpox. Viral envelopes, though, are lipid-based envelope. It may be on the outer side, outside of actually the capsid, and it may be basically derived from the lipid of the host cell. Without an envelope, a virus is classified as naked. With the lipid envelope, it is defined as enveloped. This is a very important classification, as it can play a role in the neutralization of the viruses via detergents and disinfectants. P-plomeres are the spikes of glycoproteins that protrude from the virus or viral envelope. These play a role in viral binding to specific host cells. It is these spikes that can be detected by the host immune system and thereby mount an immune response against the virus. For example, influenza viruses have spike proteins that can mutate. Two major types, hemagglutinin, which is designated by HA, and neuraminidase, which is designated by NA. Now, they shorten this if we're going to talk about the type of virus it is. For example, H1N1 is designated for swine flu. Derived from the swine, it is an influenza virus. And there are different designations for both H and N. So you can have H5N1. That is the designation for bird flu. This is usually involved in the naming convention for types of influenza viruses, as you probably presumed here. Here in this diagram, you can see the capsid. And what literally happens is the capsomeres externally form this kind of rod-like shape. But the ends of these capsomeres are attached directly to the viral genome. In an icosahedral capsid, you have the viral genome on the inside, and you have this combination of hexagons and pentagons arranging sort of in somewhat of a roundish shape. Okay, these are all the capsomeres. Finally, in a complex structure, such as what you see here in this electron micrograph that's been colorized, these are basically bacteriophages. Now, you have a capsid up here composed of capsomeres. The viral genome is inside. But what you also see are these unique structures on the outside. This is a sheath. This is a base plate, and on the base plate extends tail fibers, which play an important role in both attachment to the bacteria, and also they will contract in part as the sheath contracts. The pins attach down to the bacteria cell wall, and through this structure, it will literally inject inside the cell its genome. Now, if we look at enveloped versus naked, basically naked, there is no lipid outer membrane. There is just basically the capsid. It may contain spikes, as you can see here. And inside, of course, is the viral genome. This is an example that you would see for an adenovirus. All of these are transmission electron micrographs. With an enveloped icosahedral, you have, of course, again, the capsid and the viral genome, but you have this outer layer of lipid in which embedded into the layer is these spikes. The spikes will act to attach to the specific cell that this virus is going to infect. In this case, you see here is a herpes virus. And finally, here is an example of an enveloped helical virus. Now, on the inside, you have the helical capsid with, of course, the viral genome. On the exterior, you have the cell membrane, formerly cell membrane, just now the viral envelope with spikes attached to it. These spikes will allow for adhesion and entry into the host cell. This is actually a model, and what you see is a real example here of an enveloped helical of an Ebola virus. Now, when we get to influenza, there's one very important point which I'm going to show you in a second here. I want you to note that this is an enveloped virus. It has two different spikes here, hemagglutinin, that's the HA, neuraminidase, that's the NA. But if you look inside here, you are going to find the helical capsids that surround the genome. In this case, the genome is not a circular loop, nor a linear loop, but actually you might call it segmented. In other words, you add up the 1, 2, 3, 4, 5, 6, 7, 8 of these helical capsids, and you have the full genetic blueprint for this virus. But these segmented sections, and each individual might be called a cartridge, if they mix together with another virus, with another cell that's been infected with a similar virus, you can get new combinations here because as the viruses are reproduced, some of these will be exchanged for other viral cartridges, and you will get now new characteristics. This will lead to viruses with new combinations of neuraminidase and hemagglutinin. As I mentioned a minute ago about viral genomes, they encode capsomere proteins, various enzymes involved with viral replication, and structural factors. Basically, what you look at when you look at a viral genome is a very simplified system that's focused on either the structure of the virus or the replication mechanisms necessary. Now, viruses don't have organelles. As again, I've said this, they have to use the host cell's organelles and other structures to replicate inside the cell. A viral genome will range somewhere from a little bit less than 300 genes to maybe a little bit more than that. But here's one point to keep in mind. The larger the amount of genes on that viral genome requires a larger genome and thereby a larger virus. Okay, so the larger the genome, the more viral products are required, and the smaller the genome, the simpler the viral design and less products are required for the viral replication. Viruses can have either an RNA or a DNA for their genome. Here you can see the virus and you see the capsid inside here. This is an envelope basically. The genome arrangements. Usually with DNA, viral genomes, it's either going to be a circular loop or a linear loop. Many times it will be double-stranded. There will be other times you will have a single-stranded DNA for the genome. For the viral genomes made of RNA, they can be either linear or segmented. In other words, these multiple fragments add up to basically having the entire genome. Many times RNAs are going to be single-stranded, but there are some examples of viruses that are double-stranded. As I've said, you can have single or double-stranded for DNA. The designation there would be dsDNA or ssDNA. Viral genomes can also be in a single linear or circular molecule or multiple segmented sections, such as what we saw earlier with influenza. If you have a double-stranded DNA, it is very easy to replicate and make gene products. But if you have a single-stranded DNA, then first what you have to do is make a complementary strand of the DNA. In other words, if you have one side, you're going to use that side as the template to make a complete double-stranded. And then the gene products can be made. Now with RNA, it can be single-stranded or double-stranded. When we talk about RNA genome types, there's a couple of different classes that you want to be mindful of. First one is RNA genome functions as a messenger RNA. This is referred to as ssRNA+. Examples of this are polio, rubella, West Nile, and dengue fever. RNA genome that is complementary to the messenger RNA is basically calling it a negative strand, so that would be ssRNA negative. The genome is read, and basically you're going to make up some copies of the RNA plus, the messenger RNA, by an RNA-dependent RNA polymerase. That's RD-RPS. Now, Examples of this include the influenza virus, measles, Ebola, and rabies. An RNA genome that makes DNA, so you have to have a reverse transcriptase as an enzyme present, which is then made into the messenger RNA. So the DNA is inserted into the host cell nucleus to make messenger RNA. So that's confusing, just to help you to think about it for a second. You have the RNA genome, you've got to make it into a DNA. The DNA will then migrate into the cell nucleus, and then it will kick off lots of these messenger RNAs that will be for structural proteins. And eventually, they may include some of the RNA that's going to be part of the virus as it gets kicked out. Okay, it'll be part of the RNA-based genome. Example of that is the human T-cell leukemia virus. There's type 1 and type 2. And if that's not been confusing enough, be patient. It gets a little bit more, but not much more. You can have a double-stranded RNA. That's a dsRNA. Now that's transcribed to make messenger RNA. The RNA is unwound to make the messenger RNA. It requires an RNA-dependent RNA polymerase. The example of that would be rotaviruses. Now, as you can see here, basically, if it's DNA, you're going to have to make some RNA anyways because you have to make certain cellular proteins. If you start off as an RNA viral genome, then it depends what is the next fate. So are you going to use this single-stranded RNA to be the messenger RNA? You have to make copies of it instead to achieve a messenger RNA. Are you going to reverse this into making it into a DNA fragment and then make your messenger RNA? Or do you have a double-stranded piece of RNA and then you're going to start using that as a template to make the messenger RNA? Now, viral genomes change over time. RNA polymerases, which copy RNA, do not have proofreading, that is, error correction capabilities, that you'd see with DNA polymerases. In other words, you're going to have the possibility of having some changes occurring in the genome of this virus. These changes can be mutations in each round of replication. The reassortment events can also contribute to viral evolution. So you have two different viral strains that infect the same host cell at the same time. The genomes can mix and generate new strains. Now, one way that we're going to get a new strain is if we end up with an attenuated virus. Now, these are with some viruses with mutations that lose the ability to cause disease, yet it can still be infectious. Attenuated viruses are sometimes used to develop vaccines against more infectious strains of the same virus. Here you have viral strain A, here you have viral strain B. Notice the segments here. These are in orange, these are in black. If these two viruses infect the same host cell at the time, take over and begin starting to assemble their genomes, begin to assemble the viral capsid proteins, etc., as they're starting to be made and extruded out of the cell, you will have a virus with new genetic combinations. Part orange, part black, part orange, part black. Therefore, that can cause changes to the host range, to the trophism, to the tissue range, etc. It's very common to happen with influenza viruses. Now, with viral mutations, they can also allow the virus to escape the host immune system detection, to broaden the host range, as I mentioned earlier. So from what was previously a bat, now bird, pig, human, or even expand the type of cells or tissues the virus infects. That's trophism. So it could formerly have been a respiratory virus, but now it infects heart muscle, brain tissue, digestive system. or it can make the virus easier to transfer from one host to the other. The viral genomes can change due to two major formats. One is called antigenic drift, the other is antigenic shift. Antigenic drift, this is due to the spontaneous point mutations resulting in minor but detectable changes in the viral genome. Think about it as just a single point mutation, but as a result it changes an amino acid, and that may be an amino acid change that occurs on a protein such as the spikes, for example. Now, an antigenic shift is different. This is due to broader genetic reassortments, such as different strains of the virus infect the same host cell, especially with segmented genomes. The new viral strain can have new features, increased infectivity, and expanded host range, etc. So antigenic drift, think of drift as very short, very slow. It's like a raft drifting down a river. Minor changes, but what can happen is that one or two of these minor changes may occur. And now what was previously when you had immunity, now you do not. This accounts in part for why we have to get influenza shots each year. They look at the up and coming new viruses, they've changed a little bit, and now we make viral vaccines that are different each year. Antigenic shift is much more profound. Let's say that we have a crossing of a human influenza and an avian influenza. And now what it does is it reproduces now in a pig. The new combination comes out, and it is a highly virulent human strain. This is where we have the possibility of a pandemic if we do not have any immunity against this new strain. How do we start identifying and labeling and defining viruses? There are diverse features that are used to classify and name viruses. There is an International Committee on the Taxonomy of Viruses, ICTV. They have set up sort of the rules. Viruses are grouped by the following properties. First, the type of nucleic acid, DNA or RNA. Second, what is the capsid symmetry? Is it helical, icosahedral, or complex? The presence or absence of an envelope. The genomic architecture, is it single-stranded or double-stranded? Is it single-stranded DNA, single-stranded RNA, double-stranded DNA, double-stranded RNA, etc.? Excuse me. Also, we have additional features that may be used for the naming, such as the virus size, the host range, the tropism, that's the type of cell or tissue the virus infects, as well as disease features. Now, before you look at this and sit there and get very concerned, I'm not asking you to memorize this entire chart. Don't worry about this. If you had a virology course, that might get interesting. But I want you to take a look at this and see how it's broken down. These are just focusing on, just mind you, the medically important DNA virus families. When I say medically important, the only ones that we're focused here are human. There are a variety of other ones that infect insects, shrimp, bees, beetles, plants, fungi. Yes, there's quite a few out there. And we're just focusing on the ones that infect humans. And as you can see, you break down into viral shape, whether they're enveloped or naked, the genomic features, and then the families are broken down to what types of diseases these cause. The same thing over here. Now, host range, specific species or collection of species that the virus can infect. That's what we mean by host range. We've also talked about tropism. Now, there's different ways of saying tropism. It could be narrow or broad. A narrow tropism is basically where the virus targets only one or two cells or tissues of the host. A broad tissue tropism is where they target a wide range of host cells or tissues. Virus sizes vary. For example, rhinoviruses, polioviruses, are roughly about 30 nanometers. Now, when we talk about nanometers, remember, nano is about a billionth of a meter. The Ebola virus is different. It's 1,000 nanometers. And when we get to virus naming rules, we exclude things like domains, kingdoms, phyla, and classes. Only order, family, subfamily, genus, species, and common name are really available. And I'll direct you to table 6.2 in a second, but let's take a look at the size for a second. Here's an E. coli, okay? That's 2,000 nanometers long. A human red blood cell is 8,000 nanometers in diameter. A pithovirus is one of the largest ones that's been identified. It's only 1,500 nanometers long. A Pandorovirus, about 1,000. The Ebola is about 1,000. It's 970 nanometers long. But then you get the real small ones. Look at a bacteriophage, T4, 225 nanometers. Poliovirus, 30 nanometers. Rhinovirus, 30 nanometers. HIV, 120 nanometers. The pictures that you see here are red blood cells covered with viruses, okay? Here are the naming conventions for the viral taxonomy. You do not have to memorize this, okay? Just seeing how we go from one particular order, then we go down into family, subfamily, genus, species. The species and the common name may be similar, but may be very different. It depends. For example, you have human herpes virus 1, also known as herpes simplex virus 1. And a lot of times, the common name is there, but we don't use capitalization as much. Now, we've talked about viruses hijacking host cell machinery to multiply. Viruses acquire the raw materials, the amino acids, the lipids, the ATP, etc., as well as the ribosomes from the host cell to replicate. Bacteriophages have a means to infect and reproduce inside of the bacteria host cell. And we have to be mindful of the lytic replication pathway. This includes the 5-step process, which you'll see in the next slide. This is how viruses infect, overtake everything, and basically begin the process of replication and eventually escape. Bacteriophages will encode for lysozyme, and this will lead to killing the bacteria host cells as the viruses burst out. This is what you would see, it's sort of bacteria cell lysis. You want to also note during the process that early viral proteins include DNases. Now, why would you want to do that? Because what you're going to do is you're going to degrade the bacterial DNA. So you have a DNA degrading enzyme that basically will knock out the bacteria's chromosome so that there's no more resources dedicated to that. Okay? Here we have the lytic replication cycle. First stage is attachment. Second stage is penetration. Now, in penetration, the phage injects the genome into the cell. The protein outer structure here with the capsids stays outside. Then we have replication. This is where you start taking over the host cell machinery. The enzymes, you hijack all of the amino acids and ATP, etc., to start building components that make up the next generation of viruses. Also, at the same time, you're going to destroy the bacterial chromosome. Assembly, you start putting together all of the pieces that make up the capsid and also the genomic information that has to be encapsulated inside of that capsid. Finally, you will release all of these new offspring, we'll say, of the viruses. And how this is done is by creating some materials, some enzymes that will basically break through the cell wall and you have a bursting of the cell, it lyses, and the new phages are released. So attachment, penetration, replication, assembly, and release. Now, lysogenic replication occurs in lambda phages, and these bacteriophages are called temperate phages. We'll get into this. What is a lysogenic process? It uses the first two steps of viral infection, attachment and penetration, but then the phage DNA integrates into the host genome by forming a prophage. Now, this is sort of like hiding out for a while. The genetic information for the prophage is present. The genetic information to make this bacteriophage is present. And it can be here for quite some time. As a matter of fact, during bacterial cell division, the prophages replicate along with the bacterial genome. But if that host cell is stressed, such as exposure of UV light, nutrient depletion, etc., then the phage DNA leaves the host cell genome and becomes active again in viral replication. So steps 3 through 5 become active. When we talk about phage conversion, a prophage DNA that can incorporate inside of a bacteria can provide new pathogenicity factors to the bacteria. In other words, if you remember before, we had that transfection that can occur where we bring in some of the pieces of the phage DNA and along with other particular genetic components. Well, this is where you can have bacteria that have toxins or other pathogenicity factors. So attachment, penetration, and then we go into this integration because the phage genome adds to itself, inserts itself into the bacterial chromosome. Cell division, every time you replicate, the entire chromosome, including the prophage, is replicated. Then something triggers the lytic phase again. So the lytic cycle will trigger the prophage chromosome elements remove themselves, and this is usually when the host cell is stressed. And then what happens is you start going through replication. You will destroy the bacterial chromosome. You will begin replication of all the other components. You will have then assembly, and then you will have release. Now, this is very similar to what you see with animal virus replication. You have attachment. This can occur via capsid proteins or spikes, specificity of binding to specific host range, and tissue tropism. Penetration. This is the entry. Envelope viruses enter via endocytosis or membrane fusion. Uncoding. This varies by process and location. Basically, the idea is that you're going to have endocytic enzymes break down the capsid, so you're going to have the genetic information released. Replication, otherwise known as synthesis. DNA viruses ship DNA genome to the nucleus, so you can start making messenger RNA. RNA viruses use the RNA genome to direct synthesis in the cytoplasm. Retroviruses, that is RNA genome, direct DNA into the nucleus. Finally, we have assembly. Now we're going to start building. We have all these new pieces, so we've got to get the assembly line building new viruses. The capsids are built and assembled around a nucleic acid genome. The envelope viruses require viral proteins in the envelope before viral release to coat with the envelope lipids. Okay, so what happens is some of those spike proteins will now start inserting themselves into the lipid membrane, ready for what will be release. Release, you have the newly assembled envelope viruses released by budding off from the host cell membrane. But what if I don't need an envelope? That's when you basically have the naked viruses and they're going to rupture the host cell for release. As you can see here, attachment, penetration, uncoding, the replication process, capsid proteins, genomes, spike proteins. Notice that at assembly, what happens? If you're going to have envelope proteins, envelope viruses with spike proteins, they're going to be directed to go and implant themselves around into the host cell lipid membrane. You start forming these capsids with the genetic information inside, and they start attaching here and bubbling out. Now, assembly here is roughly the same with enveloped, but release is basically where you literally have lysis of the cells so that they're all released at the same time. And here you see HIV budding off from a white blood cell. Now, some animal viruses have unique replication mechanisms that cause persistent infections. A productive infection is where the viral entry of the host cell triggers a virion production. This causes an acute infection. And persistent infections, the strategy is to avoid the immune system clearance and yet remain in the host. Persistent viruses tend to remain in the host for long periods of time. The viruses integrate their genome into host cell chromosome, and they form a provirus. Isn't that similar to what you dealt with the bacteria, Faj? Yes. But this is also something that helps explain why you have individuals with chronic diseases that don't go away overnight. Chronic persistent exhibits a steady viral production that increases over time. Two examples, the papillomavirus and HIV. Here we have a viral replication. You have an acute non-persistent infection. Viral continues to replicate for a bit and then starts dropping down because the host immune system battles it and eventually eliminates the virus or the virally infected cells. With a latent persistent infection, now this timeline may be days, months, or years. You will have a peak amount and then you will have it drop down, but still be somewhat present in the host cells. It will have another peak and then drop down, and then over another period of time we'll have another peak. This is a latent persistent infection. Comes and goes. Chronic persistent infection, what you have, and this time course could be days, months, or years, is a very slow buildup that eventually has a slope that may be very long if you look at from here to here, and then it will achieve a higher number as time goes on. So what you have are some very interesting situations here that are requiring us to understand that sometimes the virus infection will hide for a while and then come back later on. Some viruses will actually stay in a state of hiding in certain ganglia or certain cells for a long period of time, acting as a reservoir until something triggers them to start coming back out. Okay? Here's the issue about the provirus. And this, in part, was why HIV was such a difficult challenge, because it was not just a matter of, oh, we'll treat it, we'll stop replication, we're cured. No, because it's hidden now as a provirus, and then there will be a trigger that will initiate viral RNA genome to be freed, and then viral replication, and then beginning to bud off from the host cell again. Now, a couple of terms to be very familiar with. Latent persistent. Patients with this exhibit bursts of viral replication with intermittent periods of absence. During the flare-ups, the virions are shed and the patient experiences symptoms. The virus retreats to a period of inactivity and the virion levels drop off and may be difficult to detect. Stressors may trigger flare-ups, including infection with another pathogen, fever, sunburn, hormonal level changes, and immune suppression. For example, human herpes virus 1. It will lie dormant in nerve cells, then reemerge to cause cold sores. Varicella zoster virus, HHV3, causes chickenpox, but later on in life can cause shingles. Here you have a cold sore. Persistent infections that lead to cancer can also have an explanation here, because you will have viruses that cause cancer, and they are called oncogenic viruses. Oncogenic viruses are believed to cause 10 to 15% of all cancers. The viruses stimulate uncontrolled cell division and/or host cell responsiveness to death signals. For example, individuals infected with human papillomaviruses, the strains of HPV 16 and 18. You will also have human T-lymphotropic viruses, HTLV1 and 2. They form a provirus for more than a decade before emerging to cause lymphoma or leukemia. So here are some of the examples. Again, you don't have to memorize everything, but you do need to start being aware that these things can occur. You have the examples of human papillomavirus, leading to cervical, oropharyngeal, anal, and rare vaginal and penile cancers. Human herpes virus number 8 can lead to Kaposi sarcoma. Now, Epstein-Barr virus. It also causes mononucleosis. It can associate with a number of malignancies. It's now been associated with a number of malignancies to include B and T cell lymphomas and Hodgkin's disease. Human T-lymphotropic viruses. Now, these are different. The other ones have been DNA. This is now an RNA. And this causes adult T cell leukemia. The last two, hepatitis B virus, which is a DNA-based virus, can lead to liver cancer. Hepatitis C, it has a different nucleic acid, but can also do the same thing, lead to liver cancer. Now, viruses can be grown in a laboratory. Basically, what happens is this. You have bacteriophages. They can be grown with specific bacteria type that it will infect. So basically, you set up a bacteria, you might call it a lawn, where you spread out evenly bacteria culture onto a solid agar. You let that grow for just a little bit of time, get set up, and then you do a plaque assay. Now, this is where you need to understand what a plaque is. The technique for determining the quantity of bacterial viruses, as well as how much of a given animal virus is present, uses this plaque assay. A plaque is an area that after the virus has basically destroyed those bacteria, or you can use instead human cells or any other type of animal cell to test this virus, you will end up with sort of a blank spot there. The blank spot represents cells that have been destroyed by the presence of the virus. The virus will go in, infect, release its new viruses. They will spread out and attack the neighboring cells, and they will continue to do this as they spread outward. This is an assay that can be used to purify specific viruses. The plaque-killed zones, where the viruses kill the bacteria cells, that's a plaque. And more plaques mean more viruses. Now, plaque-forming units, this is a designation called PFU. That's the quantity of bacteriophages derived in an initial volume of a sample. Here's where I'm going with this. If you don't know the concentration, the number of bacteriophages you have in a sample, one way to do this is take the sample, put it into a plate, and note the number of plaques that form. It is also possible that you can make a determination of whether the one sample of virus is very, very virulent and others are attenuated based on the number of plaques that are occurring. Here you can see the plaques. These are the clear zones. What you've done is you take your viral stock, you go through sequential dilutions, and you add this to a bacteria, and basically you have the melted agar, and you pour it into a plate. And as the bacteria, the phages destroy the bacteria. The reason why I pause is because it's equally capable to apply this same principle to, let's say, animal viruses, etc. The main point is that with these little clear zones, these plaques, you can make a determination. Each plaque started off with one virus, and they started spreading out. Now, this gets us into some terminology here. A viral titer is the quantity of virus in a given sample. Plaque assays can be used to purify specific viruses. There are some limitations. Remember that bacteriophages need specific bacteria to attack. It is worth noting right here, just stopping for a second in a sidebar, that some bacteriophages in the latter part of the 20th century, Russian scientists had tried to develop bacteriophages that, since the bacteriophage was specific only for bacteria and for specific types of bacteria, they were trying to develop, in essence, viral therapy for bacterial infections in humans. This has gone in sort of a stop-start manner for well over 70 years. The advantages would be immediately apparent since we have so many bacteria out there now that are resistant to many different types of antibiotics. The uncertainty is that no one is 100% sure whether the bacteriophages would have adverse effects on the humans. Not just from the basic perspective of, well, if I inject the bacteriophage, it's not going to affect human cells. That's true. But what if the bacteriophages cause an allergic reaction to the patient? That's not good either. So they're still doing research on this with the hope of developing phages as a means to counter specific bacterial infections. Now, animal viruses grow in only specific cell lines. If the cell line doesn't exist, a viral culture may require a live animal instead. Viral cultures in specific animal species is also required. Live viruses are necessary for the development of diagnostics, antiviral drug development, and vaccine development. Now, what you see here, you see the development of these plaques. You see the blank areas, the dark areas starting to spill out. All right, let me explain to you what you're looking at right now. I was able to get this as soon as the news item spread around January 30th. On January 23rd, this virus from a patient at an Australian hospital was extracted, and they basically started to find a means to culture it outside of China. So what you see is a video of the coronavirus, its growth and forming plaques on the plate. And the plaques demonstrate that cells are destroyed from the viral replication. And why everyone was so excited about this, see, we're going to start again here, was because knowing that they have a cell line that they can reliably grow the virus on gives them the beginning of an understanding of how to culture the virus, how to start developing drugs that would interfere with viral replication, etc., by just using plaque assays. Let's move on. Now, a lot of times today, what we do is we use for viral culture embryonic eggs, chicken, duck eggs, things like that. There is a challenge here because some small amounts of egg protein may still exist if we are using this viral culture to develop a vaccine. This can thereby induce allergic reactions to patients allergic to eggs. Why do we do this? Because if you can culture the virus to a larger number, then inactivate them using something like formalin, etc., you have an inactivated virus that you can use as a vaccine, a type of vaccine. Now, Some more recent research has been using insect eggs, which offer promise for viral culture without the allergic reactions. Here you see, if you're going to cultivate influenza virus or mumps virus, where they will inject into the allentonic cavity. The allentonic cavity injections for influenza virus, amniotic cavity for the influenza and mumps, allentonic cavity for culturing influenza virus, mumps virus, adenovirus, and then yolk sac injections for HHV, that's human herpes virus. Here you can see someone holding up an egg, lighting it up to get specific location of the areas for injection. This is an influenza vaccine called FluBlock. Of course, here we have a little chick. There are certain facilities that all they do, they monitor eggs, they incubate eggs, they harvest the eggs, they inject the eggs, etc., day in and day out. Now, diagnostic tests determine the presence of certain viruses. Remember that we have to cultivate the viruses so that we can eventually start developing the antibodies to develop diagnostic tests. Various tests exist to determine the presence of the virus or that the immune system has battled the virus. Basically, we're looking for antibodies. Case in point, chikungunya virus. Because the virus is pretty much swept out of the system within 9 days after initial infection, the only way that patients may be reliably detected post that 9 days is by the presence of antibodies. And most clinically useful tests require the following: specificity and sensitivity, as well as turnaround time. Specificity, that is where the diagnostic test reliably detects only that virus of interest with no false positive results. Case in point, dengue fever, Zika, and chikungunya are somewhat related. Early tests had cross-reactivity when it came to making a Zika test. The Zika virus test, if you remember, if someone gets ill with it, if the child is pregnant, it will end up with a smaller skull size. Okay, and what happens is that when they were testing individuals in different countries, they were finding that they were testing positive, but it turned out that it wasn't Zika, but it was that they had already been exposed to dengue fever or chikungunya, and in some cases even just yellow fever, which were very endemic in those areas anyways. So you've got to basically have a reliable test that doesn't give you false positives. Sensitivity. The test detects low levels of the target to limit false negative results. You can have a sub limit, a very low limit, and you can still be infected and you can still be infectious. And if the test misses you, you've got a false negative. Turnaround time. Ideally, tests should have a very short amount of time that is required for receiving results. And so this is extremely critical for any diagnostic test. Some tests basically would take a week or longer. Now, of course, they take much less. And with COVID-19, the initial rush to make tests was that they had to be able to be reliable and they had to be sensitive and specific, but they had also to be quick in a turnaround. And what that meant was you take the test, you take the sample, and in some cases, test kits are being made now where they basically are able to demonstrate a definitive answer within 15 minutes of initiating the test. Now, in some tests, we wanted to detect viral proteins. So this is where you have situations. The term antigen is proteins that antibodies target or bind to. With immune assays, that is the antigen antibody-based tests, test kits that use antibodies to detect viral proteins. one issue that you've got to keep in mind is you've got to find the antibodies that are going to be effective, selective, and very specific. They have to be very, very sensitive to even small amounts. Now, there are different types of tests. The agglutination test. This is where the antibody-coated beads, upon mixing with the sample, whether it's blood, saliva, urine, you will get this agglutination or this clumping up in the presence of the virus. So what happens is that the beads have antibodies attached to them. The antibodies will bind to the virus, but other antibody-coated beads will also start clumping up until you get this clump, this agglutination present in this type of test. Now, the lax agglutination test uses viral protein-coated latex beads, which will bind to viral antibodies. But then we also have the enzyme-linked immunosorbent assays, otherwise known as ELISAs. And these can detect viral antigens or antibodies. The ELISA detects viral proteins or antibodies and can change color if there is a binding or a clumping. Limitations: the antigens and the reagents must be well characterized. If the virus undergoes any antigenic drift or antigenic shift, that kit may not be able to detect the virus. Also, the patient needs time to seroconvert. Now, what I mean by seroconvert, this is related to the term seroconversion window. This is the time period after infection for the immune system to develop detectable levels of antibodies. Problem: it can be weeks or months later. Seroconversion for chikungunya may take some weeks. A lot of times it will be after the virus has been eradicated, and this makes it difficult for some individuals. Another case, HIV. HIV, the virus would still be there, but it takes up to 6 months from the time of infection for an individual to develop antibodies to HIV. The first tests in the mid-80s that were first made were looking for antibodies to HIV. Problem: they missed the first 6 months. So someone who wanted to get tested came up false positive, thought they were safe, and may still infect others until later on they subsequently had some of the the opportunistic infections, or they had another test that showed the presence of the antibodies. Here's the situation I was talking about. For example, you can have the beads coated with antibodies. They will bind to the virions, and then you have this clumping up that occurs because, note, for each virus, you may have multiple antibodies on multiple beads attaching, forming this clump. Now, you can do it the other way around. You can have the viral protein stuck on the bead. You're looking for antibodies from the patient's serum, and as a result, you get the clumping there. Here you have an agglutination, you have a positive result. Here you have no agglutination, a negative result. Now, what about really cutting through all of this and just looking for viral genetic material? Hey, viral genetic material, the detection methods are very powerful. They may be also much, much more sensitive. Therefore, you don't have to worry about, when the antibody showing up or anything else. Okay. They can also, in a lot of cases, if you get the right DNA targets, you can be able to identify new viral strains as well. Now, several different techniques exist. One of the best is polymerase chain reaction, PCR. Also, you can use fluorescently labeled probes. The concept is to locate the specific unique nucleic acid sequence of that virus. That's it. Now, what about treatment? Antiviral drugs treat infections, but do not typically cure them. This is because that viruses can become a provirus and have latent periods. Many drugs will block active viral replication, but will not go after latent provirus presence, basically the nucleic acid presence. Now, there are a variety of mechanisms of action for selective antiviral drugs. They can be attachment blockers, penetration blockers, uncoating blockers, replication or viral assembly blockers, viral release blockers. There is also the post-exposure prophylaxis. This is a preventative treatment applied after exposure to limit the infection. There's several examples of this. One of them is human rabies immunoglobulin. And basically, if the person has been exposed to possible rabies, what they do is they then give that as injections. It used to be 30 days in the stomach each day. Yes, you heard me, in the stomach area, in the abdomen, for 30 straight days. Now they think that they've made some improvements on it, so it's only like 3 times over a period of a month. And I'm not sure, but I think they still have to do it in the stomach. Very painful, but it beats getting rabies and dying. Here are some of the examples. Do not worry, I'm not asking you to memorize these, but you want to get somewhat familiarized. And this chart should be hot paged. Why? Because later on we're going to be talking more about antiviral drugs in the antimicrobial drugs chapter. Okay? All right. Now, how do we do it? Well, for example, nucleoside analogs. These are analogs to nucleosides which block DNA polymerase, RNA polymerase. Example would be acyclovir. We have also antisense antivirals, the short sequences of nucleotides that complementary bind to the RNA transcribed by certain viruses. Double-stranded RNA is then destroyed by cytoplasmic enzymes. We also have interferons. These are released by cells as a signal to the immune system that the cell is infected with a virus. It's used to treat select viral infections. The problem is it tends to cause flu-like symptoms, but it has been known to treat hepatitis B and hepatitis C. Here it is an example of acyclovir. Notice that it is very similar in structure, except for this area right here, to guanine nucleotide that is normally added to the DNA. So what you end up having is the machinery stops by the presence of this particular product. All right, now we're going to move on to prions for a few minutes. Prions have been really interesting, and if you ever get to read some work by some of the people that played key roles in its research, including Stanley Prushner, who eventually got the 1997 Nobel Prize, you'll find that it gets to be very, very interesting and in some cases very much a challenge to scientific thinking. Prions are acellular. They lack nucleic acid. They are infectious proteins. Well, wait a minute. Don't you need nucleic acids to make the proteins? Well, not exactly. They do not replicate as they don't have nucleic acids. Then how do they replicate? They multiply by distorting similar proteins. These misshaped proteins come in contact with normal proteins and twist the protein to a misshapen state. The proteins will clump together in nerve cells and lead to brain tissue degeneration. That's where you get into what's referred to as spongiform. They, a lot of cases, cause a class of diseases called transmissible spongiform encephalopathies. If you're not familiar with encephalopathies, you'll probably, if I say to you, mad cow disease. Now, for humans, we have Creutzfeldt-Jakob disease, CJD, Gershman-Strauser-Schneiker syndrome, which is a inherited form of prion-based disease, fatal familial insomnia, which is another inherited one. And then we had mad cow disease. This is also known as bovine spongiform encephalopathy. Now, in Britain, in the 1990s, there was a massive outbreak of a particular disease. And let me give you a little bit of a history of it quickly. There was a disease called scraping, that sheep would start getting into this very scraping against posts and things like that, but it became self-destructive. They continued to do it when they had lost all the wool, and then they would do it and continue to keep moving back and forth till they literally had open wounds. So the sheep were considered waste, and they were rendered into protein pellets. You see, in Europe, how they basically had protein supplements for their cattle is use what they had referred to as offal, or basically OFFAL, basically using renderings from other animals. After the mad cow incident, that was all stopped. But basically, the prions that caused scrapie passed over into the cows. The cows had mad cow disease, where basically they would have a hard time standing up and moving around, and they would be listless and everything else. And it turned out that their brains were deteriorating. Unfortunately, the British loved their beef, and some of this meat was contaminated and led to what is called variant CJD. And this was due to the consumption of prion-contaminated meat. Oh yes, even if you cook the meat, it does not go away. Also later on, it was found that it can be transmitted by cadaver pituitaries. Why cadaver pituitaries? Because the pituitaries were taken from cadavers, and they would extract the growth hormone that would be used for growth hormone shots for short kids and things like that. That was found to be a big mistake, and they stopped that. Iatrogenic CJD. This is caused by dura mater grafts, transplants, corneal transplants. These have been caused by the medical procedure. In one case, with contaminated stereotaxic surgical equipment that was used to treat individuals with brain diseases, it was found that they could not decontaminate correctly and sufficiently. So the stereotaxic surgical equipment basically passed on prion-based disease from one patient to 13 others who eventually died from the same disorder. Now, in the West, particularly up in the Rockies and areas like that, you have animals called mule deer and elk, and they are facing a similar prion-based disease called chronic wasting disease. And the interesting thing about that is this is a pathogen that's active, infectious, and can be let out by a sneeze, by saliva, by urine, and by feces, or by consuming mule deer or elk. And so this has caused a great concern. By the way, to help you to understand something, prions are resistant to breakdown. They've actually done studies where they've taken a culture with prions, buried it into the soil for 2 years, and it came back up infectious. They actually did a study where they found an area where scrapie had occurred. They left the pasture land fallow, meaning no activity, for 18 years. And when they put sheep back in there, they got infected. Because you got to remember, when sheep consume, if you've ever seen them consume grass, they consume grass with some dirt particles, etc. So it's been found that prions can remain active and infectious even in the soil. This is not good. Here is a normal brain, and here is the spongiform. You can see where a lot of the cells are dead, and they begin to start spreading outward. You have these big holes, just like a sponge. Here's a staining for these in neurons. The red is all of prion accumulations. The next thing I'm going to show you is computer program, and I'll show you that in a minute, of actual prions infecting the other protein and restructuring them into a prion-like state. But there are two major theories of how prions reproduce. First is that you have endogenous, and by the way, PRP stands for prion protein. PRC is the normal prions, or normal protein, I should say. PRPSC is scrapie, in other words, the infectious disease-causing prion protein. So what happens is that you have some of the normal proteins come in contact with a scrapie protein, we'll call it that, scrapie prion. They will go through conversion, and the conversion will eventually lead to more of them beginning to accumulate, and eventually what you have is many of the proteins going through this reproductive cycle here to eventually build up and lead to eventually the death of neurons. The other point is this. You have infection where the normal proteins get converted into prion, scrapie prions, and what happens is these start forming these long structures, and they break off into what they would call new seeds and infect other cells. But basically, these long structures tend to eventually block up the normal function in the axons of the neurons and lead to the death of the neurons. As I said, the first hypothesis tried to explain how prions replicate in a protein-only manner, and this was what was called the heterodimer model. Okay. Now, the alternative model assumes that the scrapie prions exist only as fibrils, and that the fibrils ends bind to the new untouched proteins and convert them into scrapie proteins. So there's several different theories on its development. The main point is that these are infectious. As a matter of fact, if any nerve material in international trade, for example, in Asia, they import a lot of US beef. If any of it, even though we've kept prion diseases out of our food stream, if any of it shows up, they stop importation immediately. Any country that announces that they've had a case of scrapie, mad cow disease, something like that, immediately undergoes an embargo. And it may be a long embargo before they are able to start having trade again with beef. This computer animation, here you see beta pleated sheets. Okay, that's a sort of structural arrangement of the protein, right? Here you have alpha helices. Now what you have is a prion influencing the normal shape and structure of a healthy protein. And as you can see, this is through a computer animation where they were doing analysis of this, and it's reshaping the entire structure. And you're going from alpha helices to just a structure that is basically composed of beta pleated sheets. And this is now a completely infectious prion. Well, we've pretty much finished up on this chapter. I encourage you to go over the visual summary on page 189, review the chapter overview on pages 190, as well as the end of the chapter questions on 192. Don't forget to do your homework this week, which is due at the end of this week on Friday, 5 p.m. Excuse me, not for 5 p.m., but it's due, the homework is due by 11:59 on Friday evening for Mastering Microbiology. The quiz is due by 11:59 p.m. on Saturday evening for Mastering Microbiology. Take time to review the videos in the Blackboard for the lecture content. The next area we will go into is Fundamentals of Microbial Growth. That's Chapter 7. These are just a couple of other images that I wanted to consider using. I hope you have a very nice day.